His name is Research Professor Mahmoud ElSohly, and he has been since 1980 the director of the only National Institute of Drug Abuse financed and approved marijuana farm in the United States. So the professor has done an extraordinary amount of research. It's a 12-acre marijuana farm. I told him, Professor, you were going to bring samples, but I guess they didn't let you that on the plane, huh? But he's going to talk to us a little bit about his research and what he thinks about the medicinal benefits and the allegations by the proponents and the opponents to marijuana, and is there any medical benefits to it? So it is my pleasure to introduce you to you now, Research Professor Mahmoud ElSohly.
Okay, well, good morning again. Okay, I'm going to, my name, as Mr. Bishop said, Mahmoud ElSohly. I am the research professor at the National Center for Natural Products Research at the University of Mississippi School of Pharmacy, and I'm the director of the marijuana project at the university there, which is funded by the National Institute on Drug Abuse. And I'm here to pose this question that's on the board. Marijuana, it's a very complex botanical substance or product or plant material. Can that be actually brought to the point of being a prescription drug? So, I'm not biased one way or the other. You probably by the end of my talk, you will see really where my thinking is. But I think I want to take you through the whole process, the botany, the chemistry, the products, the manufacturing, the growing, all of that stuff, and then take you through some of the activities that the plant might have or the compounds in the plant that are known to exist and produce the biological activities might have, and then decide whether we really have something that we can approve as a drug, or we still have a lot of work to do before we can get to that point. So I'm going to leave answering that question to the end. In the meantime, I'll also bring you or share with you some of the products that come out of the plant material that we can actually develop into true pharmaceutical drugs to deal with the activities that we are seeking in this particular plant. So, to get started, let's talk about the species. Marijuana over the years has been, people talk about it. It comes first of all from a plant known as the cannabis plant, and that cannabis plant, cannabis is a genus in the plant kingdom, is a genus that belongs to the family Cannabaceae. It's that family, the name comes from the cannabis plant. And then it used to be thought that the genus cannabis has 3 different species or different species: Cannabis sativa, Cannabis indica, Cannabis ruderalis. And then in 1976, we had the United Nations Narcotics Division come to the University of Mississippi, where I was actually there starting in 1975. So I've been there 40 plus years now. But they came in 1975 to study or to talk about the botany and the chemistry of the cannabis plant. And out of that conference, that's an international conference, came the consensus of the scientists around the world that the genus cannabis is actually a single species, which is Cannabis sativa, but then that species is highly variable, so you can see too many varieties within the cannabis plant. Variety sativa, variety indica, variety ruderalis, and within those, there's a lot of hybridization. It's a very highly variable species, meaning you start out with one plant and you have other plants around, and before you know it, the seeds coming out of that plant produce materials that are a little bit different than the mother plants, and so on. So it's a very highly variable species. But having said that, the plant itself or the different varieties could be classified as different chemotypes, and those different chemotypes are based on the chemical profile of the plant material and the major components, the major cannabinoids. Cannabinoids, as we will see in a minute, are those chemicals that are very specific to that plant. They have never been found or discovered in any other plant in nature. That's what makes that plant a very, very special plant. And so we have three different major classes. The drug type, which is high in THC content, the fiber type, which is high in CBD content, and intermediate types that have a little bit of both THC and CBD. But then on the market, you can see a lot of different products. The most important and most widely used is the plant material, which is marijuana, which now people call medical marijuana. And then you have the hemp seed oil, which is actually taking the hemp seeds and pressing those to produce an oil that's an edible oil with very, very low level of THC or CBD, simply because the seeds themselves don't have cannabinoids. The cannabinoids are contaminants on the surface of the seeds as a result of physical rubbing against the leaves when they process the seeds. But then you have the hemp oil, which is actually the extract of the hemp plant, which has, depending on the type of plant material you start with, they will have CBD, mostly CBD, but they also have some THC to some degree. And then you have the CBD oil, which is basically the same as the hemp oil, but is produced from high CBD varieties of the cannabis plant. Then most people, or some people, or maybe most of you here that are not very familiar with the plant, they don't know that the plant actually exists as a dioecious plant, which means you have male plants and female plants. And the myth out there now is that the male plants don't have cannabinoids, they don't have THC, which is not true. The male plants do have quite a bit of THC, it's just not as much as the female plants. And also the biomass is not as much per plant as the female plants, and therefore in the illicit market, you'd never see male plants being marketed. The other problem, of course, with the male plants is the male plants have pollen. The pollen produces allergy, and therefore people try to avoid using that, especially if they're going to smoke it and take it into their lungs. That can cause some problems, allergy problems. But you can see on the left side, the male plant, very skinny, very skimpy, and then the female plants are nice and plumpy, and they have a lot of flowers and good amount of biomass per plant relative to the female plant, to the male plant. Then also, I mentioned earlier that we have the fiber type plant, the fiber type variety, and the drug type. The fiber type is the one on the left, which is just produced for fiber, and the drug type is produced for the drug quality of the plant, which is the content of the cannabinoids. Now we know something about the plant and the botany of the plant. Now we get into the chemistry. This is, as I mentioned, and as the title of the talk says, is a very complex natural product. There are right now, our last count, we have over 546 different compounds that have been identified, characterized, detected in the plant material, and that makes it a very, very complex natural product. Each one of those compounds have their own biological activities, and whether it produces a high or produces a low, increases the blood pressure, reduces blood pressure, increases the IPA. There are so many interactions between those different compounds that you have in the plant material that in order to really have a drug out of that, you gotta really know what you're dealing with. You gotta know what the composition is. You gotta know what the chemistry is. But anyway, make a long story short, the plant is very complicated. You have a lot of different compounds. And if you're going to be smoking this plant, smoking this material, each one of those compounds is going to break down as a result of the extensive heat. The temperature of a burning cigarette is 700 or to 800 degrees Celsius, not Fahrenheit. And therefore, it's a very, very hot. And therefore, the compounds will evaporate and get into the smoke, into the lungs, but also it will break down, not just evaporate, but break down. And each compound will produce several breakdown components in the smoking process, and therefore, by smoking a cannabis cigarette, you are literally ingesting thousands of chemicals into your body, and maybe the benefits that you need to have could come out from one or two of those compounds that you are ingesting. So, very complicated natural product. Now, one of the classes of compounds that exist in the plant material that is very specific to the cannabis plant is known as the cannabinoids. The name of those compounds comes from the cannabis. Cannabinoids. These are compounds that exert the majority of the biological activities that the cannabis plant is known to produce. This table here that you see, we have now identified a total of 104 different cannabinoids. Delta-9 THC, the compound that everybody is familiar with, is just one of those cannabinoids. CBD is just another cannabinoid. And then we have all the others there. And in the plant material, when you smoke a cannabis cigarette, you're ingesting all of those, the interaction between all of those. What you're getting is actually the net biological activity of the interaction of all the compounds that you are ingesting. Now, having said that we have 104 different cannabinoids, the ones on the board right now, and most of you probably are not chemists, so I'm not going to elaborate too much on the chemistry here, but suffice to say that out of the 104, there is only 6 cannabinoids that exist in large enough concentration to really make a difference in terms of the overall biological activity. These are the ones on the board here, Delta-9 THC, which is the major main psychologically active compounds in the cannabis plant. THCV is a sister or a brother of THC, but it just has a shorter side chain. And then you have the CBD, which is, everybody is familiar with CBD now, and CBD oil and all of that. This is another compound that has its own biological activity that's different, or maybe share some with the THC, but majority are different. And then you have a compound called CBC, cannabichromine. That compound is known to have good anti-inflammatory activity and good antimicrobial activity. It's got the pharmacology of its own. Then you have CBG, which is cannabigerol. This compound is known to have very, very good antimicrobial activity. So we have a variety of different compounds with different pharmacological properties. Then the last one there, CBN, which is cannabinol. This is actually a degradation product of Delta-9 THC. It has activity about maybe 20% that of THC. So these are the major cannabinoids that we find in the plant material. Now, I'm just going to go through those slides very quickly. Just suffice to tell you that when I talk about the Delta-9 THC type structure, the plant material is rich in so many different components that relate to that. In the plant material, the THC that we all are familiar with does not exist as THC, as the free THC. It exists as a precursor, which is known as the THC acid A, which has a carboxylic group in the 2 position, right where this says R1 right there, or in R3, you have a carboxylic group there. That carboxylic group, that compound, the precursor that exists in the fresh plant material, does not have psychological activity at all, does not have much of a pharmacological activity. In order to get the pharmacological activity, it has to be decarboxylated, meaning removal of that carboxylic group to produce the free THC. And that free THC is the one that produces the psychological properties and all the pharmacology associated with it. That decarboxylation process happens by heat, and therefore that's why marijuana is smoked. You cannot take fresh marijuana, bring it in, and just eat it as a salad. You're not going to get high on that at all. So it has to be heated either by smoking or maybe cooked in a brownie or a cake or something like that. So it has to be subjected to heat in order to produce the psychological properties. Then we have the Delta-8, which is just an isomer of the Delta-9, has about 80% activity of Delta-9, but nonetheless exists in the plant material in very, very small amount, not much at all. Then the cannabidiol, this is the one that everybody is interested in these days for the treatment of epilepsy, but the same story holds true, meaning it exists in the plant material as a precursor. that has to be heated or dealt with in order to that carboxylic group and produce the free CBD. Now, there are some work, or there is some work going on right now to establish whether what we call cannabidiolic acid, the one with the carboxylic group, does actually have pharmacology similar to that of cannabidiol, but the jury is still out there on that issue. But we know that cannabidiol itself does have very good anti-epileptic or anticonvulsant activity, and therefore it would be a good drug for the treatment of epilepsy. And this is not really something new. Back in 1975, when I joined the University of Mississippi, I went to my first meeting on cannabis at the University of, at the Research Triangle Institute in North Carolina, and that's where I first learned about the anticonvulsant activity of CBD. But no one moved with this, no one took that and tried to develop it because there were so many other compounds available for the treatment of epilepsy, and therefore getting something out of the cannabis plant back then was really not a good thing to do. Pharmaceutical companies were not interested, so it never went anywhere until the last 2, 3 years, 4 years, when there was a renewed interest in cannabis and medicinal properties of cannabis. Then we have these other compounds, which is degradation products of CBD and THC. And then now we're taking all these compounds and all these products that we were talking about and looking into the illicit market, what's out there in the illicit market, and we see all different types of preparations. And those include those kilobricks that are classical materials coming from Mexico. Then you have the sensimilla, which is the buds coming out of the female plants of the cannabis that have not been pollinated from male plants, and therefore there are no seeds. Sensimilla is a Spanish word for no seeds. And then you have the buds, which are just seeded marijuana flowering heads. Then you have the Thai sticks, which is an old preparation that comes from Thailand. All other different products, hashish and hash oil, and come in different forms that exist in the illicit market. You can see some more examples of those products in the illicit market. And we have done analysis. We have been getting samples from seizures that are made by the DEA ever since I started back there in the early 70s, and doing what we call the potency monitoring and looking at the trend in the THC concentration, the potency of the cannabis preparations on the market. You can see the graph just climbing all the way. And I just started there from the 95, but before that it was even lower than that. But anyways, you can see the trend is an increased potency over time, and it's been now plateaued since 2011, 2012. And reaching about, on the average, 12% or 13% delta-9 THC in the plant material. Now, these graphs represent the other cannabinoids that I'm talking about, and I put it on a separate graph from the delta-9 THC because their concentration is so small that if I put it on the same graph, the previous graph, then it's going to be just down there at the bottom as one line. You can't really see much. But this is here, you can see on the right side the percent, and so they are all below roughly 0.5%, 0.2%, 0.3%, and there is no special trend in the change in the concentration of these other cannabinoids over time. So the only one really that's changing so much is the THC. CBD, there are some changes that are going on, and I show you that in this graph. So now we're talking about delta-9 THC and CBD as the two major cannabinoids of interest to everybody today. And there has been some talk about why the National Institute on Drug Abuse in the production of materials for research only focus on high THC content plant material and not high CBD. There are some interest in doing the pharmacology of plant material that has a combination of CBD and THC. So I went back and looked. National Institute on Drug Abuse, of course, their mission is not to develop drugs for treating any disease in particular. That's the charge of other agencies within the National Institutes of Health. But the National Institute on Drug Abuse is charged by looking into the abuse potential of different drugs and what do we do to curb that abuse and to treat that abuse of these or addictions to these drugs. So NIDA, National Institute on Drug Abuse, they were interested in to monitor what's going on in the illicit market and try to produce preparations that will mimic what's on the street so that they can do research or investigators can do research with materials similar to what's out there. In looking at this graph that you see in front of you right now is looking at the ratio of delta-9 THC to CBD in confiscated materials, not in any pharmaceutical product, but in confiscated material. And you can see that over time, the ratio keeps climbing up, meaning that people on the street in the US are seeking products that have high in delta-9 THC, the drug quality, the high quality of products on the street. And you can see that now it is just sky high now, the ratio, meaning that you can see plant material on the street that is maybe 20% delta-9 THC, but only maybe 0.2% or 0.1% or 0.3% of CBD. That makes the ratio of THC to CBD 60 or 70 times more than CBD. So the trend on the street is high THC content. Now with the interest in the treatment of epilepsy, children and so on, the picture is changing a little bit, not necessarily on the street material, but on some of the products that you see being sold for that purpose on the internet. So I just want also to take you through the cultivation practices for this type of plant material, just to get really some idea about the differences that could exist between different products that you might see on the market. We have, of course, the indoor cultivation, outdoor cultivation, micropropagation, biotechnological, processes, and so on. This is our grow room at the University of Mississippi there, where we actually prepare materials before we take them out to the field. And these are cuttings that are ready. The ones on the left, on the top shelf on the left, these are the mother plants. And we, in order to keep the genetic material stable, we have to propagate what we call the vegetative propagation from the mother plants so that the genetics and the chemical profile of the products coming out would be the same day in and day out. Because if you start from seeds, as I mentioned before, it's a highly variable species. If you have a bunch of seeds even from a single plant and you grow those seeds, you're going to get different plants of different chemical profile simply because the plant is genetically variable. But in order to keep the genetic material so you have the same chemical profile, you have to start from either vegetatively propagated plants or micropropagated plants so that you can have the same genetics. These are plants that we have grown indoors in our grow room. Female plants, of course, all coming from cuttings. Very nice looking plants and buds. And you can see that with the hydroponic system, you have a tremendous root system there. And then these are the cannabinoids that I talked about, the 104 different cannabinoids and the terpenes and most of the biologically active compounds in the cannabis plant are produced on hairs, glandular glands that exist on the surface of the leaves of the plant material. And the smaller the leaf is, the more density of those glands and the higher the THC content and the cannabinoids content. And therefore, if you look at a big plant going from the outside into the core of the bud, the THC and the cannabinoid content goes up. So you can have one plant, and within that one plant, you can have actually different products. If you want low potency product, you take the large leaves. Medium, you take the medium leaves, and high potency, you take the small leaves or the bud. These are the bracts that surround the female flowers of the cannabis plant. This is a electron microscope magnification of how those glands look like. You can see that the head of that gland is where all the cannabinoids exist. Tissue culture, we have developed some ways of doing tissue culture, direct organogenesis. Usually when you do tissue culture, you have shoots that come out that are maybe have different genetics. They just mutate in that process. But we have developed a system or a protocol that will keep the genetics from changing. So all the shoots coming out of that tissue culture will have the exact same chemical profile. And we have taken this all the way from the beginning, all the way to getting them outdoors, putting them into the soil and so on, and looking at the chemical profile and looking at that and show that there is genetic stability in the process that we have developed for that. Then we have indirect organogenesis, where you can actually have a callus from a leaf tissue, and then that will develop later on into shoots and different plants. Then we have developed what you call a synthetic seed. This is basically to keep the genetic material, to keep, if you want to have produce the same type of plant, the same chemical profile every year, you can't do it from seeds. You have to do it, you have to keep it in a way, either through continuously vegetatively propagating the plants or through the micropropagation, one of those techniques that we have here. And those synthetic seeds, the one on the top left, is nothing but some apical nodes that you can encapsulate and keep it, preserve it, and then later on you bring it out, you put it in you let it come to life and then produce, like you see here, the succession from A to B to C to D, and all the way taking it out into putting into a pot. And that plant that comes out there will be exactly the same as the mother plant from which we took those synthetic seeds to start with. When we say synthetic seed, we don't mean they are synthetic, they are natural, they're coming from plant material, but they are preserved like seeds, but they're really not seeds. They are, plant tissue. Then we did looking at the chemical profile and the changes in the chemical profile over the growing cycle of the plant, between the vegetatively propagated plants versus the mother plants versus the micro-propagated plants. And we can see the black, the white, and the shaded. These are the three different types, and it depends on the stage of the growth. You can see that all three look exactly the same or very, very, very similar. No change over time. Then we did also the genetic, looking at the DNA of those, and we can see that the genetic makeup is exactly the same, no changes. These are the plants before we took them outdoors, and here is our outdoor garden. Mr. Bishop told you that we do have a big outdoor field, 12 acres field, but we don't grow 12 acres every year. Last time we grew was 2014. We had 1.2 acres. And the one time before that was 2007. So we really don't grow as often as people think. We don't produce as much material as people think. There is, we have the capacity, but the need is just not out there. 2014, we produced 800 kilos of plant material. I still have 780 kilos, so there has not been that much demand on the plant material. So people are yelling and screaming and hollering, saying, well, neither doesn't have this, neither doesn't have that, but we can't just give it to the people. They have to ask for it. They have to have a protocol, what they're going to do with this plant material, and we're happy to produce whatever people need. We also took some of those plants in 2014, put them in hydroponics outside, and look at the plants. They look so nice and so pretty. And so you can do it either in the soil, or you can do it in the hydroponic. Either way, you're going to get a good product. The genetics is very similar. The chemical profile is very similar. It's just a matter of how much material you need to have and how the capacity that you have in either operation. Of course, outdoors, you can produce a lot more. Here's again our field in 2014. Here is the plant material getting a little bit more close to maturity. And then here is the harvesting of the plant material. And then we take that plant material. These are the buds that are harvested from after drying and so on. You can see these are, very, very big buds that have also a good, so biomass-wise, needless to say, we can produce a lot of material. So the idea that we cannot produce enough to cover whatever the demand is, that's not really true. We have a lot of capacity for doing this. This is the processing, the drying of the plant material. We have a drying barn, like a tobacco drying barn. That is all temperature controlled, time controlled and everything, and then we process that. And then we have a machine that we ask the company to make specially for us, because this is called a de-seeding machine. De-seeding machine, usually you do de-seeding because you want to get the seeds. In our case, we don't want the seeds. We want the plant material. So we had to reverse engineer the process so that we can get the plant material and not get the seeds. And all the way at the end over there where that cyclone is behind, I think I'm the guy with the blue shirt there. Right behind me is a cyclone where the leafy material comes in. It's allowed to settle and then comes down into that liver pack barrel down there where the material is collected, and you can see it here, in this slide. Very nice, clean material, no seeds, no stalks, no contaminants in polyethylene bags that are FDA approved and all of that. All our operation, by the way, is Food and Drug Administration approved. We have a drug master file on this material. It's registered with the FDA as a GMP manufacturing, so it's all done as a true pharmaceutical preparation. Then we have the products that you get out of the plant material. You got the plant itself, which can be used for any chemical investigation, any investigations that not necessarily in the form of cigarettes. Then we make cigarettes out of that for the national program, for investigators that are looking into doing studies with the smoked material. Then we have an extract, that barrel that has the tar-looking material. This is a cannabis extract. This is what you extract out of the plant material, very complex material. And we have developed a process to isolate the individual cannabinoids out of that big mesh over there. And the one on top, the white crystals, is a derivative of delta-9 THC. And delta-9 THC is really not a crystalline material. It's a very unstable. It's just a very difficult chemical to work with. But we developed a process whereby we can convert the material from the extract into crystals that we can keep for a long time, and it's very stable. And then at the time when we do need THC, then we can take those crystals and do one-step chemistry to convert it back to the THC, which is in the flask on the top right hand side, which is very faint yellow-looking material, very viscous, very thick, which is the delta-9 THC. Now, in 2014, because of the need for medicinal materials with the high CBD content, we were charged by the National Institute on Drug Abuse because of the demand of the scientific community to produce material with high CBD. In 2014, we produced, we put in some new cultivars. We have 5 different cultivars of the CBD-rich species. These are the top 5, and then we have 9 different cultivars that we have genetic material to use for future production that have both delta-9 THC and CBD, and then we have just one high THC variety that we have been using, and the National Institute on Drug Abuse has been having in their drug supply program for many years, and that's the one on the bottom. Then if you take the high CBD material and you do the extraction, then you end up with a product, the extract, that dark green material that might have as much as 40 or 50% CBD in it, and then depends on what solvent you use for extraction and how the extraction process goes. Now, so that takes you through the chemistry, the production, the manufacturing, and so on of the different products that you get out of the plant material. Now, this plant material does have some, it does have biological activities. It does have possible medicinal values. And so let's look at that in terms of what we call the cannabinoid receptors. Cannabinoid receptors, these are receptors in the human body that respond to the ingestion or the use of the drugs or the compounds that you get out of the cannabis plant. The cannabinoid receptors. There are several receptors. The list is big, as you can see on that slide, but I'm going to only refer to 2 cannabinoid receptors that are very common and really can account for the majority of the activities that we are seeking to use cannabis or cannabis preparations or products for. And these are the CBD, the cannabinoid receptor 1 and cannabinoid receptor 2. CB1 exists mainly in the central nervous system, but then also in the peripheries. But then the cannabinoid receptor 2 is existing mainly in the immune cells and in the peripheries, but there are some indications that also exist in the central nervous system, but not to the same extent. So, this here I got a couple of few complex slides a little bit, but just want to have them there to show you that those cannabinoid receptors, there are what we call affinities for those receptors. And on the left side, we have the different cannabinoids, and then on the right side, we have what we call the binding affinity. And you can see, if you can see, actually read it from there, I can't, but if you can read it from there, that the THC binds much better to the CB1 receptor and the CBT receptor, but the CBD actually does not. The difference is almost 1,000-fold. Somewhere around that the affinity of the CBD to those receptors is very low, and therefore CBD actually does not have much of a psychological activity at all, and that makes it really a good drug. If it has a good biological activity that we can take advantage of, then it's good, because it does not have the side effects of the producing the high and getting into the central nervous system. Then we also have what we call the endocannabinoids. The body, our human body, does have compounds that act just like THC. And it's regulated by it produced in the body, by the human body, but then also on demand, if you will. And then once the body doesn't need it, then there are some compounds that are called fatty acid hydrolase enzymes that will break down, those endocannabinoids, and therefore you get the activity is removed. So we build endocannabinoids and break endocannabinoids depending on demand. If we are under stress, then the body will produce endocannabinoids that will make you feel maybe a little bit better, will make the situation a little bit better. But then once the stressful situation is gone, then the body will break down those endocannabinoids, so you cannot stay high, if you will. So, the function of the endocannabinoid, I'm going to skip that for now, because that's a little bit more into the a little more basic questions here. The receptors have potential modulation of the receptors. If you stimulate the CB1 receptor, what happens? If you block the CB1 receptor, what happens? These are the, modulating the cannabinoid receptors is what ends up to be what causes the compound to be used for a given indication or the other. Depending on what you have a modulator, you have something that will increase the activity of that receptor or diminishes the activity of that receptor. You have pharmacological activities of that. So I'm going to skip with that one and just go directly to what would happen from a therapeutic standpoint. If you stimulate, agonists means stimulate. If you have a CB1 agonist, that's a compound that stimulates the CB1 receptor. What happens when you do that? This is like THC. You can see here, you can use that activity therapeutically for the treatment of nausea and vomiting, wasting syndrome, which is appetite stimulation, in other words. And then you can use it to reduce the intraocular pressure and therefore use for glaucoma, or use for multiple sclerosis and spinal cord injury, because it does have a protective effect on the on the nerves. And then if you antagonize, if you have a compound that antagonizes or suppresses the CB1 receptor, that compound could be used for the treatment of schizophrenia, could be used for treatment of obesity, or could be used for the treatment of drug abuse. In other words, if you give a person a CB1 antagonist, a person that is craving, marijuana, that's addicted to marijuana, you can suppress that demand, you can suppress that quest to have more, THC or more marijuana by giving this compound. However, this compound here, there's a compound that was produced a few years ago by Sanofi pharmaceutical company for the treatment of obesity, but then they found out that it does have, because it suppresses has a negative effect on the central nervous system, there was some side effects that were undesirable, and therefore they stopped the drug. So you really need something that will have not too strong antagonistic activity to the CB1 receptor, because then people were getting suicidal by using this compound. So it's a very tricky situation there. Now for the CB2 receptor, if you stimulate the CB2 receptor, you can use it to treat pain, inflammation, and cancer. And some of the compounds from the cannabis plant actually have very good anti-cancer activity. But for antagonizing or diminishing or suppressing the CB2 receptor, you can use that for the treatment of arthritis and autoimmune disorders. So it's a balance. And one thing that I failed to mention in the beginning, which is very, very important, and that is those cannabinoids have what we call biphasic activity. What does that mean? It means that at a certain dose, certain concentration in the blood, you have one biological activity. At higher concentration, you have the opposite. You don't keep getting higher and higher and higher and more pleasant activity by using more and more and more THC. No. If you use THC at a certain level, you're going to get high, you're going to get pleasant activity, you're going to get hungry, the munchies and all of that. But then if you use more, then you're going to get the opposite. You're going to get dysphoria. You're going to get irritable. You're going to go crazy. So it's a very tricky balance, how much drug you really need to have in your system in order to get the right biological activity. So we can say, right now, these people have 30% THC in plant material, 20% and so on. You cannot consume a cigarette out of that without dropping, not necessarily dead, but dropping meaning because you had too much that you get into the narcosis, and then when you wake up, you're irritable, you're paranoid, you just had too many side effects. And we can see right now, as I showed you on the graph that showed the continuous rise in the THC content, that as a result of that, or maybe not necessarily as a result, but coincidental with that, the rate of emergency room admittance as a result of, with the cannabis use as a precursor to getting to the emergency room, is very, very much parallel. If you see the rise in the number of emergency room admittance post-cannabis use versus the increase in the THC content over time, they're parallel. So, people say, well, that may be coincidental. Maybe it is. But it's certainly, it just makes you think and wonder why such high THC content preparations on the illicit market. Anyways, so now we're getting very close to answering the question about the cannabis. Could it be a prescription drug? So in order to answer that question, I'm going to take you through the process of what we talk about in botanicals in general. We talk about botanical drugs versus dietary supplements. A dietary supplement is something that's out there on the market that, there's so many dietary supplements, herbal dietary supplements that are there, and the manufacturers of dietary supplements claim that they would help the general health of the body for this and that and the other, but they cannot claim any specific treatment, mitigation, or prevention of any particular disease. That's a dietary supplement. A drug is, or a botanical drug is, medicine plants, extract thereof, that, or components that are isolated from medicinal plants that are used to treat, mitigate, prevent, diagnose a particular disease. So there's a big difference between the two, and we're going to go through the process for. So in order for cannabis to be marketed as a medicinal agent, as a drug, as a prescription drug, you got to go through the process that I have there on the slides. You got to be manufactured under CGMP, good manufacturing practices, with all requirements of quality assurance and quality control. You got to guarantee quality of the final product. You got to have consistency of the product from time to time to time, any time you prepare it. And the chemical composition, absence of microbial contamination, heavy metals, pesticides, all the rest of it. You got to be clean. You have to prove the safety of the product. It has to be proven. The very product that you are marketing, not just a generic marijuana, is okay. No, the product that you are making, you got to show that your product is safe. You got to show that your product is effective. Then you got to go through defined protocols and documents for having that product. Product information, you got chemical properties, physical properties, label information, certificates of analysis. You got to do all of this. You got to do it in the operation. You have standard operating procedures. You have record keeping. You have master drug file for the product that you are producing, batch production records for the product that you are using, drug master file that you file with the FDA that contains all the specifics of the product that you are putting there. Your personnel have to be qualified. The FDA facility has to be registered. I mean, your facility has to be registered to the FDA. The physical plant and grounds and environmental assessment information has to be provided to the FDA in order for the product to be classified as being manufactured under GMP. You got to go production process, flow charts, detailed description of the process. You got to go processing products, control of materials, control of critical steps in the process, and quality assurance production. I mean, the list goes on and on and on in terms of what you have to do, validation of the process, validation of the analytical method that you use to quantitate or qualify your material. And then you have to have standard reference materials for the product that you are producing. Then you have the packaging. Everything has to have specifications and labels and all of that. Then you have to have the quality assurance in production, stability. You have to put your product on stability. The product that you're putting on the shelf, how long it stays on the shelf without being decomposed or degraded or changing the potency enough that you cannot sell it anymore. You've got to have some specifications there. You have to do all of this in order to put that and get it approved by the FDA. So there are some products here that I've just showed. I got that courtesy of the Families in Action in Georgia. They got me that these are products that you have that are called medical marijuana. You don't see a packaging there. You don't see a package insert. You don't see instructions. You don't see anything. You just see a product there. And you just walk there and you just pick what you want. Oh, this one here looks really nice. Oh, this one here has a good flavor. A flavor is nice. I'm not against flavor, but flavor is not going to treat your disease. They don't tell you how much material you have to use. They don't tell you how frequent to use it. They don't tell you, how much material you need to treat a particular disease that you have. All these conditions have to be met in order for that to be a prescription drug. Now, here are some products on the market. I obtained all these products and analyzed them. With the exception of one, maybe two products. Out of all these products there, they didn't have much of anything. They really did not have much of anything. Very, very low levels of CBD and very low levels of THC. So they're just selling stuff to the people, but don't really have any value for that matter. And the ones that have high amounts of CBD in there, but they don't tell you what the dose is supposed to be. This is the famous Charlotte's Web. And Charlotte's Web does have actually some CBD in it, but again, the dose is not defined. It's almost like trial and error. You just take it and try and see what will work for your child. That's not a medicine. That's not a prescription drug. Anyways, states have legalized marijuana, but they haven't really done much in terms of, anything in terms of, doing what needs to be done to make it a medical marijuana. And then, in terms of the medical association, a lot of medical associations have opposed, 3 minutes? Okay, have opposed, medical marijuana issues, but there are some products on the market today that are actually being manufactured in a GMP. Marinol, Sesamet, Sativex, Epidiolex, all these are true pharmaceutical products that have gone through all the process that I mentioned to you earlier. And then here is just a comparison between legalized medical marijuana medicines versus the FDA-approved drugs. In the legalized, you have no purity, no informed consent, no randomized clinical trials, no safety, no efficacy, no recall system. Doctors certify, but they don't actually prescribe per se. Dispensaries sell, and bud tenders dispense, as opposed to FDA-approved drugs that are pure, informed consents, randomized clinical trials, safe, effective, recall system in place, doctors prescribe, pharmacists sell, and pharmacists dispense. So there's a big difference between what you call medical marijuana today and what FDA-approved drugs should be. I have a lot more slides, but I know I'm running out of time. So I would just, 1 more minute. Okay, we have developed some new products, and these are suppositories with that actually produce really, really good biological activity. I know people here in the US don't like suppositories, but it's very much acceptable in the medical community and in other countries. And we have developed transmucosal delivery systems that you can put it above the gum line and get the benefit of the cannabinoids. And we have also produced eye drops for glaucoma, and we have shown that you can actually reduce the intraocular pressure by developing medicines that you can use in the form of an eye drop in the eye that will lower the intraocular pressure and treat glaucoma. These are true pharmaceutical products, not just something that people say, oh yeah, this is medicinal marijuana. So come back to answer the question about medical marijuana. Can marijuana be a prescription medicine? The answer to that question is yes, it can be, but it's not today. Not the way it is being put on the markets today, because it has not met all the requirements. If you can have a manufacturer that will take a product and go through the approval process, go through the quality control, quality assurance, safety, efficacy, dosing, and that dosing has to be specific to the disease you're trying to treat. If all of this done, then yes, for sure you can. But today, medical marijuana as it exists in the different states that have passed legislations to do that cannot be a prescription drug. I don't see how you can have that. Anyways, I'm more than happy to answer questions. I told Mr. Bishop that I really wanted to talk more, but he said, no, you got 45 minutes, but maybe you can go a few more minutes more, but not more than that. So I'm sorry, I have to shut up. Thank you so much for listening to me.
Professor, this was outstanding. We really, really appreciate it. I know that Representative Harrell's got a question for you, but for us to keep on time, well, I'll tell you what, we'll take one question. Go ahead.
Okay.
Thank you very much, Bernie. My question deals with the medical research. The double-blinded studies that any pharmaceutical medication that is under prescription must go through the entire 4 stages of medical research, going through double-blinded studies to show both the efficacy, the side effects, the safety of it. Has any of those medical marijuana products gone through and proven to the same level of efficacy that penicillin has to prove to be effective?
Well, the answer to that, obviously, not. No, they have not. There's not, like I said, no manufacturer, grower, or producer of any medical marijuana in any state has actually gone through any of those processes. The first thing, if you want to come up with a new drug, like even the drugs that we are producing right now, which actually come from the cannabis plant, but we are taking those and then modifying those to make them real drugs, even though it's coming from cannabis, and cannabis is being used everywhere and everything, we have to go through the same process. We have to take that and do the first thing is a phase 1 clinical trial to show safety. The first thing is safety. We have to show the safety. And then after that, you do the phase 2 clinical trial that shows maybe dose ranging for a particular disease. Not a blanket like medical marijuana is. Medical marijuana is just, you can use it for anything of this disease, that disease, everything. With the real marketing of a drug, you got to do the clinical trials for what indication. For example, Marinol, which is the delta 9 THC in a soft gelatin capsule, it has only 2 indications on it. And that is, the first one is for nausea and vomiting of cancer patients getting chemotherapy, and for AIDS patients suffering from the wasting syndrome as an appetite stimulant. Anything outside of those 2 indications on the label is what we call off-label prescription. So in order to really get approval and be able to put in the label that is used for this indication, you gotta do the clinical trials for that indication. So none of those products on the market have done that. I mean, on the medical marijuana, have gone through any of that process at all. None of them is registered with the DEA, none of them is registered with the FDA, so it's just Thank you.
Sir. Let's give them another round of applause. We really appreciate you being here. Thank you so much.